How brain evolutionary mechanisms could inspire artificial intelligence structural designs is mm, a tremendous issue, and I don't want to disappoint you, but to be honest, I do not work directly in intelligence, but I would like to share with the audience some results we have from morphological developmental genomics and behavioral studies, and in the context of studies from other researchers as well. If you have any question, please let me know. Uh, so my presentation is going to be organized a little bit like this. I'm going to talk about homology, about cerebral cortex evolutionary development, also about gene co-expression and functional network analysis, and some insight about networks and artificial intelligence. But before that, I would like to show you this. This is actually an activity that we perform with our students. Uh, so here we can see a very, very simple network formed by 5 neurons. Uh, and actually, every student is one of these number of the network of the neurons. The neuron number 1 is one student, the 2 is a second, a third, a fourth, and a fifth students, and they are working interactively. So for what we are doing this is because we think that doing this, we can introduce some concepts that are more elaborated, more complex or abstract for them, so they can be the neuron and they can see how uh, different connections will have a specific weight that are these squares with numbers over the arrows. Yeah, there are directions of influence, and in this case, this network is multiple connected. So every neuron is connected with every all other neuron. So we can see here the synaptic contact that are uh, showed by the edges of the arrows. And also every neuron have an activating threshold, which is the square inside of the circle. Uh, and the initial states, in this case, for example, the neuron number 1 is in white color because it's activated. The number 2 is off, is inactivated. The number 3 is activated, is on, the 4 is on, and the 5 is off. And that is generating the first pattern in time 0. So now if we move to the table at the right side, we can see the neurons from 1 to 5 and the time. The time 0 is describing the status that I said before for every neuron. So what the students have to do with this, they have to record the influence that every other neuron has over them, and they have to compare the sum of the influences with his states, and then is transiting, is moving to the next time, to the next stage of network dynamic. And all of the students, all of the neurons, or all of the nodes, whatever you want, they are updating at the same time. So it is a synchronized network. And we can see that from time 0, they are transiting to a second stage in 1 with 0, 1, 1, 1, 1, then time 2, 1, 0, 0, 1, 1, and time 3, 1, 1, 1, 0, 0. And then we have time 4, which is 1, 1, 0, 0, 1, and then it's transiting by different uh, patterns in the number in the time 5, 6, 7, 8, 9, and 9, and then at 10, the same pattern in the time 4 is repeated. So in this case, for this network, we have this uh, different patterns that we call them like a cycle. Okay, and what we ask for the students after they are constructing their network, they are using random procedures to set the weight and the thresholds, for example, and they have to change something. Sometimes it's the structure. For example, they can change the weight or the thresholds of any neuron in the network. Other times they have to change some initial stages, for example, and some of this network, sometimes they get the same results, sometimes they change, sometimes instead of a cycle that is transiting for different stages, we go to the same stages. So for what I'm using this kind of networks, it's because every network has a structural information that is coded in the weight and direction of influence, in the number of synapses, in the thresholds, and also in the structure of the connectivity. So this very simple network allows us to introduce to the students over dynamics of cognition even. So when we discuss the risk, motivated, and we can discuss different aspects of neuroscience. This is a different network. Here we have the input, where they say the data, and we have a first layer, which is the input layer, and then we have layers that are hidden, and then we have an output layer. So in this structure, we have to present data. So it's gonna be perceived by the input layer, and it's gonna be processed by the hidden layers, and then we will have a result. And we can train this network to optimize the results, training the network to be better in the result that we want to get. So basically, the networks in artificial intelligence, most of them are associated to this, which is presenting a pattern, and we want some solution, and we would like that the network rebuild itself to create this right answer. And this is some kind of the problem that we have in evolution, because we would like to compare in brain evolution, we would like to compare different networks, neuronal networks from cerebral cortex or other nuclei, and we would like to compare them, and that's an issue because we don't have so much information about that. So in brain evolutionary development, then we have this this issue, which is a very tough question but very important. We have observation. The merging of cognitive and associative functions has been key to the success of the evolution of mammal, and in particular in humans. So it is remarkable that lacking of cerebral cortex, some birds have impressive developed reasoning and social skills. So here we can see how these birds have different cognitive uh, functions like causal reasoning, imagination, prospection, flexibility, complex cognition. So if we attribute these capacities to the cerebral cortex, so then what's happening here? Why other species can do some similar performances? So some questions that arise from this information is, for example, can different species use different networks to generate similar behaviors? Is that what is happened? So how? Because when we create a network, we talked before that we need neurons, we need connectivity, and in the brain we have modularity with compartments and microcircuits and everything. So one question here is, how can we move from an acknowledged hidden neuronal circuit to replicate complex behavior? So I'm going to talk now about evolution and genomics, how we compare different brain structures. Uh, and then I'm going to come back to the network that I showed you before, so to get some insights. So one problem that we have in evolutionary development is that we work with living species, and we want to know about what is the evolution, but the evolution already happened. So here is some situation in A that is called homology, when a feature is shared by two related species. In this cladogram, in this tree, we can see that the red star in F and G is shared, so we can say that in the smaller star, these species F and G, they share the feature, so we can say that this homologous, and this it means that the hypothetical ancestor of these two species has the feature. The same happened with E and D in the first example. All of them has the feature, exhibit the feature, so this feature is homologous. But sometimes we can detect similarity, and this similarity can be shared in example in F and G. And here we can say that this is homologous between F and G. But if we see in E, the feature is not present, and neither in D, but we can see this feature in C. So what happened here? We call this that this is not homologous, and we say that this is a convergent pattern, which means that independently, this similar feature emerged during the evolution, uh, not sharing with the ancestor of this species. So this is the problem that we have to address. We would like to know about evolution, in this case, in a cladogram from different lineage of mammals. But during the evolution of mammal, we can see that many mammals disappear, like here in the light blue color, and we have some living species that are representative. So when we want to know what is the structure that are homologous or non-homologous, we have to work with this, with comparison between living species. And this is also because the brain is a soft tissue and it's not fossilized, so we can't have much more information about the inner structure of the brain. Also, we are limited by available data. And at the right side, this kind of cladogram in circle is showing the available genomes for vertebrate species. So there is a project that is has a goal for getting like 1K or 10K genomes, and that's going to be great because then we can compare between different species, dissecting, for example, some compartment during development or in adult, and comparing across species. Okay, now linking evolution and development, we have to recognize that there is some similarity in one stage of development. And this is the hypothesis so long ago by Hegel that say ontogeny recapitulates phylogeny. So that means that in one stage of development, which is called pharyngula, we have this shared morphology that is at the left, uh, highlighted by the brownish color. So if we analyze the gene expression between species, we can see that at the early stages of development, when, for example, gastrulation is happening, we have a very high divergence in gene expression, and those genes that are expressed are more modern genes. But at the pharyngeal stages, at the middle of the embryonic development, we can see that this divergence is not so high, it's very low, and most of these genes are very preserved across the species, are ancient than other genes. And then this diversification again happened to the more adult stages or later stages. Fortunately, in nerve system development, we can see something very similar. If we compare vertebrates, here is the chicken and mouse brain at the pharyngeal stage, we can see that these species share the morphology, firstly, and then the expression of transcriptional factors that are specifically expressed in independent compartments of the brain development. So that happened during development, and what happened in the adult? Well, very early, Professor Harvey Carten described that there were similarities in the brain circuits between the avian brain at the left and the mammalian brain at the right side. Uh, Basically, these two brains are very different. Okay, so for example, the cerebral cortex that we can see in the mammalian brain in the dorsal part, that here is in colors, in different colors, we can't see that space or that region in the avian brain. And what avian brains and reptilian brains have is a huge mass of cells protruding to the ventricle, to the lateral ventricle, which is called dorsal ventricular ridge. Professor Carten observed some similarities between the cells in the dorsal ventricular ridge and in the mammalian brain. And this similarity is also at the level of connectivity. For example, here we can see some laminar columnar auditory cortex in avian brain at the right side. And this connectivity is very similar to the organization of the connectivity in the mammalian cortex, where we have some input that are processing in the intracortically in the layers, and we have some outputs descending. The same happened to the avian auditory pallium, but this is happening mainly in the dorsal ventricular ridge and not in the topologically equivalent region. So this is different in the same that we are seeing some similarities in the dorsal compartment of the pallium, which is the dorsal part of the telencephalon, and here we are seeing this in the lateral and ventral pallium. This similarity is also sharing the visual system and also in the somatosensory system. So basically what I want to say is that the dorsal ventricular ridge is receiving information from auditory input, visual input, and some somatosensory input similar to the cerebral cortex. And probably that's the associative nuclei in the brains of avian species and reptile species. But in mammalian species, is the cerebral cortex, the device that is doing the association of information of different modalities. So Professor Carten proposed an hypothesis that consider that excitatory neurons that participate in this new network are migrating tangentially to invade the cortex. In the case of mammals, we can see that in A and in C. And in B, he proposed that the same cells are migrating, protruding to the lateral ventricle, generating the dorsal ventricular ridge. That was a very good hypothesis, but it wasn't proved. Actually, when we observe tangentially migratory behaviors of cells, we can see that, but that happens only for inhibitory cells and some excitatory cells, but that are transient populations. So inhibitory cells exhibit this tangentially migrating behavior, but not excitatory neurons that are preserved in adult stages, which is the hypothesis of Professor Carten. Even more, if we compare the topologically equivalent structure across reptilian brain, avian brain, and mammalian brain, we can see that the cortex is very different. The dorsal pallium, in this case, is very different. In reptilian brain, it's very simple. It has only 3 layers, an external plexiform layer, an inner plexiform layers that are layers that doesn't have so many cells. They have projections mostly, and they have a central layer, which is the cell dense layer that has the neurons actually. So it's not complex as the mammalian cerebral cortex. And also in the correspondent region of the avian brain, we have a nuclei which is called hyperpallium. And during development, the hyperpallium has neurons that migrate readily to the surface, yeah, but only from a very restricted point in the ventricular zone, which is the lining zone that is lining the ventricle, and which is the proliferative region here in this species. And also the gradient of migrations is outside in. What that's mean? That's mean that the oldest neurons are located in the surface, and the earlier neurons are located deeper and deeper and deeper. And that is different between mammalian brain. Now, if we move to the mammalian brain, we will see that the radial migration is in an inverted pattern, especially for the cells that are going to form the cortical plate. So at the beginning, we have some earliest neurons generated that are located in the marginal zone, which is the future layer 1, and in the deepest layer, which is the subplate. So those cells are the first generated neurons, and then they are divided like a sandwich by new wave of neurogenetics of neurons that are developing. And these new neurons have the ability to pass through the subplate and the early formatted layers and be located below the marginal zone, below the layer 1. So that is creating an inside-out neurogenetic gradient. So that is different. So we can see that there are different morphologies and pattern of neurogenesis across different brains. But also we can see that the proliferative regions are different across vertebrate species. At the first, we have the turtle brain. This is a cornal section for one hemisphere, and in the middle there is a space. I'm showing the lower figure, and we can see this pink color lining the ventricle, and that is the ventricular zone, which has progenitors that are going to form neurons. But from sheep and in marsupial, there is a debate, still a debate, but in rodent and leucanid primate, we can see that other progenitors, in this case staying with blue color, are amplified. And these progenitors, we can see them in the pallium, in the dorsal part of the brain, and also in the subpallium that is going to form mostly the striatum. So we can see here how at the level of proliferative regions, we can see this more complexity in the rodent and of course in the primate brain. That could be related with the higher expansion of the cerebral cortex and more complexity. In the case of the primate and the mammalian brain, we can see the 6 layers from the layer 1, 2, 3, 4, 5, 6, and even more. Layer 4, for example, is subdivided, and the layers 6 is also subdivided. So what happened in this proliferative compartment? We can see heterogeneity between the progenitors. We can differentiate radial progenitors, which are undifferentiated stem cells, neuronal stem cells, like subtype 1 and subtype 2. And these radial progenitors can generate neurons or generate more progenitors. And the same happened with the radial progenitor 2, can generate more progenitors that can even amplify more the population of undifferentiated neurons with mitotic capacity, or can go to generate intermediate progenitors that are mostly distributed in the subventricular zone, which also can generate more neurons. So when we see this more complexity distribution of progenitors in the proliferative regions, like in blue the subventricular zone and in pink the ventricular zone, we are observing that these devices are amplified during development and across evolution. So what is going to happen in these proliferative regions? Well, these progenitors, here the cells, the neuroepithelial cells, are going to be divided symmetrically to generate more progenitors. So you can amplify the number of progenitors if you can divide them in this way, or you can have an asymmetric division, which is to generate postmitotic neurons that are going to migrate radially to the surface. And here we can see how radial glia cells can also generate intermediate progenitors that can also generate more neurons. So in this case, for the mammalian brain, we have two devices. We have the ventricular zone device that is generating more progenitors, and we have the subventricular zone device in green that is also generating more neurons. At the age of the development stage and after birth, we have the generation of astrocyte after the generation of neurons. So one question is why increasing the neural progenitors would be associated to more computational capabilities. So we have more neurons, but so what? Is that actually getting us some more complex processing? And that's what I want to discuss now. So we can see that these progenitors can be divided and generating more neurons, but these neurons are related to each other. So they form some kind of clone, and this clone, we can see that in the figure upper right, are related to each other to connectivity, cell-to-cell connections, which are gap junctions or connections uh, or fast synaptic or electrical synaptic contacts. Okay, and if we analyze the respondent behavior of these neurons to, for example, the orientation of columns, we can see that those neurons that are clonally related, which means that are derived from the same progenitors, have the ability to recognize the same pattern of column orientation. Even more, this radial migration is generating some organization that is called column. And when we can see this column in rodents, we see that this column is not so complex like in higher mammals. That is much more complex. So we can see that there is a lineage-dependent circuit assembly in the neocortex across development. And how this happened? Well, we have these ontogenetic columns, which is the one that I talked before. That is the generation of descendants neurons from the same progenitor, and this is a clone. And they will have synaptic connectivity at P1 to P6 that is driven by gap junctions, these electrical junctions. And then during the maturation process, after birth, this connectivity is going to be reinforced by chemical synapses. So, and then we will have this microcircuit that is able to process information. So if we amplify progenitors and we amplify neurons, then, or nodes for networks, we will amplify also the microcircuits. And these microcircuits, we can assume that will have a better representation or more likely representation of or perception, sorry, of some kind of information in the case of the column organization. So my point here is that if we amplify progenitors, we can amplify these computational units of processing, which are the microcolumns, and then we will have more computational capacity for the cortex. And this is happening mainly in some regions. If we compare the human brain with the brain of other primates, we can see that in this heat map, the red color is the most changing areas of the brain, and we can see that the temporal parietal region is the most highly changed at the level of morphology. It's increasing a lot. And also the prefrontal cortex. Both regions are regions that are associated to abstract processing of information, language, and different processes that are higher hierarchy of cognitive processing. So how can we explain that, or how we can control the generation of these proliferative regions, the generation of more progenitors, then more neurons, and then we can assume that they will form more columns and more processing units? Well, one model that we are proposing is that signalings from different parts of the brain in neighbor regions of the dorsal pallium or dorsal cortex in mammals can generate signals like Wnt3, that is a signal that can control the same signal, the proliferation of neurons, but also the cortical specific differentiation program. So the same signal can amplify the progenitors and also can participate in the cortical differentiation. So that can explain why the dorsal pallium is expanding. And this signal is generated by a region that later on is going to be part of the hippocampus that is called the cortical MCH. A different in sauropsids, which are avian and reptilian species, we don't have this highly developed cortical M, so the wind signal is not so high, but we have a very high signal of PAC6 deriving from the region that is called the anti-M, and that corresponds to the ventral pallium. So we can amplify this region in red in sauropsids, and from that region is generated mostly the dorsal ventricular ridge that I mentioned before. So we can recognize this neighbor signaling from organizers, but there will be also a local control in the progenitors itself. We saw some heterogeneity as well. So there are different steps that can allow us to control the proliferation and then the number of progenitors and neurons. This is a parenthesis, is to mention that the brain development is not an enclosed process. It's interacting with different organs during development. And this is one of our study about interactions between the placenta and the brain. And we can find more than 100 interaction of diffusible signals, endocrine signals, and these genes involved in these interactions are associated to processes that are very important for brain development, like dendrite branching and synapse formation, or neural stem cells proliferation, cell polarity, cell migration, invasion, neurogenesis differentiation, and neural outgrowth. So basically, what I'm saying is that we have local control, we have a neighbor control, and we have a long distance control, which is the endocrine control. So there are different organizations of brains. We can see that now, I think, but it's even more. And I'm not explaining all the details of this. These are some examples, but as I said, in the developing pallium of the birds, we can see this columnar migration from the ventricular zone, and in the mammalian developing cortex, we can see this migration to form the cerebral cortex, which are different because of, for example, the layers in this avian dorsal pallium, which is the hyperpallium, was considered that each of these HAM, HAI, IHA, IH, IDM were considered layers of processing, but it doesn't make sense because the layers in the mammalian cortex, they are generating from different position in the ventricular zone, and in the avian brain they are generating mostly from a very restricted ventricular zone, and these cells are migrating orthogonally to the surface. And also, I talk about the microcolumns and columns circuitry that are relegated to the dorsal cortex, to the cortex in the mammalian brain. In D, we can see that. But in B, in the figure B, we can see that these columns in avian brain are transiting between different brain compartments, even subpallium, pallial compartment, and dorsal pallial compartment for the visual or for the somatosensory, and also for the auditory cortex. Uh, so in this case, we can see that this functional organization is quite different, and in the avian, the processing of the information is happening in different brain compartments. So we can see this similarity, and we would like to define, as I said at the beginning, which is the corresponding part of the brain across the species. So then we can construct the evolutionary history or a scenario of this part of the brain. And there were studies that were supported by connectivity, some homology, by gene expression of some restricted genes or developing genes, other type of homology. And there was a very, very high debate, longer than, I don't know, 10, hundreds of years of debate or 50 years of debate about the correspondence between the brain areas in different species. So we ran a transcriptomic analysis, which is an unbiased analysis. We dissected different parts of the brain involved in this debate about homology, the dorsal cortex, different layers, the hippocampus, the striatum, the amygdala, the claustrum, the preformed complex as well, and we compare with different nuclei of the avian brain. We use the hippocampus and striatum as control because nobody is debating about their homology, but we also dissected the hyperpallium, the mesopallium, the nidopallium, and the arcopallium. The mesopallium in M, and the nidopallium in N, and the arcopallium, those nuclei form the dorsal ventricular ridge. So to do this, we use different kind of analysis, and one of them was weighting gene co-expression network analysis that basically is using an independent scale a process algorithm to generate models of gene expression that are co-expressing each other, so they have a high correlation pattern of expression across different areas. And we did that for the mouse brain and for the chicken brain. And then we projected statistically then to see which areas were preserved or not across the brain. And we observed that the transcriptome of the striatum is very similar to the striatum, bingo, that was our control, the same for the hippocampus, which are very, very different in the morphology, but the transcriptomes, with the transcriptome, we can see this similarity. But for our surprise, we also detect similarity between the dorsal ventricular ridge and the layer 4, which was one of the layers that was proposed by Professor Carten as homologous of the neurons in the dorsal ventricular ridge. However, this shared signature is just 5 genes, a hand of genes, no more than them, and these genes are associated mainly to the function of the neurons and not associated to developing processes. So, considering that these genes are co-expressed between the dorsal ventricular ridge and the layer 4, and these regions are originated from different developing regions, we proposed that the explanation for this was convergence. So basically, from this study, we observed diversification because outside of the striatum, the hippocampus, and this convergence of the dorsal ventricular ridge and the layer 4, we didn't detect statistically significant similarity across any other area that we study. And we observe exceptional conservation and convergence as well. And after that, there were more studies using some transcriptomic analysis, in this case, from a songbird and a human brain. And we can see that the high vocal center, for example, is very similar with the aligned human region. So there are similarities in the high vocal center of the songbirds and the vocal regions that control vocalization in humans. So going back to the network that I showed you before, I know that several modeling groups at the cellular, intracellular, and molecular levels, and even more in the complexity of the neuron, would like to know more about the molecular composition of the biological objects that have been modeled. So my question, after we can see that we can increase the computational abilities, improving the control of the proliferative regions, it is plausible to model structural parameters of large scale biological neural neurons in order to optimize complex recognition and cognitive processes. So basically, can we go for more networks more similar to biological networks? And that's my question, because at the moment we are working with hidden layers mostly for deep networks that can recognize data that we present, and that can give us an output, and that is very good to optimize processes. But can we merge these like different modularities to get a more complex recognition and cognitive processing? So that is the question: how to use the information that we have from comparing different brains and developmental processes to feed this kind of neurons. So, in summary, I was discussing about an evolutionary scenario in which convergent, divergent, and conserved developmental programs are activated in specific compartments. So, developing neurons are modulated by local neighbor and distance signals. We observed that the evolution of this developmental program in primates would account for the expansion of neocortex. And most important, a specific developmental program will allow for the acquisition of a brain with more computational capacities and advanced social cognitive functions. So I think that from this approach we can debate about how to rethink and artificial intelligence and network structural parameters can be so I would like to acknowledge to Francisco Boiti, Sultan Molnar, Grant Belgard, Heidi Kauni, and Manuel Malikeo, which all of them were working with me in these studies.
Thank you very much, Juan, for the great talk. Now it's time for questions from the audience. Any question? Oh, okay. So I would like to make your question is you speak about the developmental role or the role of development in the generation of different networks allowing behaviors, and then it comes to be the proposal of Garstan about develop the relationship between development and evolution. Instead of Hegel point of view, Garstan posing that development creates evolution. It's not a recapitulation like proposed by Hegel. Instead, it is kind of the way that is created evolution. So, in this way, thinking about Garstan, then it would be interesting if you could make any comments on the role of development in some way the rationalization and gene expression, changes in gene expression to make possible these changes.
Sorry, Marita, I'm not
No me entendí.
listening to well. I don't know.
Okay, maybe.
No, I understood,
My
but
the volume of the computer.
there was a problem with the
Okay, no, it's just a comment about.
maybe,
Okay,
but
yeah. Didn't you follow?
uh, what we can see when we compare adult brains and developing brains is that in the development stages we can see we can detect high pattern of conserved gene expression, but apparently these genes that are preserved after that they are changing a lot, and there is a functional transcriptome that is dominating the genomic progress. So what I want to say is that okay, we have this developmental preserved pattern and organization of the brain, but after that, during brain development, apparently there is a high connection with the environmental input, and that is
Epigenetics.
epigenetics. That's right. So and we can see that a lot, and our current studies are associated different exposition of intrauterine development about uh, drug drugs during development, stress during development, metabolism during development as well, and we have some information about that. But all of this uh, environmental determinants are affecting the later performance of the brain. So there is a mix of this uh, development, drive and evolution. Okay, that's great, but the development of the brain is in synchrony with the environmental demands.
Mm-hmm. Okay.
And then we can move to population evolution and selective processes, which is another story.
you.